All right, guys, today we're going to talk about chapter 20, which is the digestive system. It's a pretty long and hefty chapter, so we're going to have to spend quite a bit of time here talking about the different pieces, but hopefully some of it's not the first time you've heard it. So the digestive system really is responsible for transforming food into chemicals that cells can absorb and we can use for energy. So in a pretty simple statement, that's what the digestive system does. We're going to talk about all the different pieces of the digestive system and what they all do. So we have the main digestive tract includes the mouth, the pharynx, the esophagus, stomach, large intestine, small intestine, rectum, and anus. Basically, it's a tube that extends from the mouth to the anus. Then we have the accessory organs, and those are the salivary glands, liver, pancreas, and gallbladder. So we'll talk about all those as well. And what really happens within the digestive system is digestion, which is the breaking down of food and particles into nutrients that we can absorb. So there's mechanical digestion and chemical digestion. Mechanical digestion, which is the first phase of digestion, really is the physical breaking down of food into smaller pieces. So that's really going to occur mostly in the mouth. Chemical digestion then is digestive enzymes that break down food further into nutrients that are then absorbed through the intestines. So both of those processes are digestion. All of these organs are going to play a part in digestion. Three layers of the digestive tract. There's 4 layers actually. The mucosa consists of its own 3 layers, an inner layer of epithelium, a layer of loose connective tissue, and a thin layer of smooth muscle. The submucosa is a thicker layer of connective tissue, contains glands, blood vessels, lymphatic vessels, and nerves. The nerves here are really regulated by the autonomic nervous system. They regulate motility, which is movement of the digestive tract, secretion, and blood flow. So when we talked about our fight and flight nervous system and our rest and digest system, this tract here is controlled by our rest and digest system. Then we have the muscularis layer, consists of 2 layers of smooth muscle. An inner layer encircles the tract and the outer layer runs longitudinally, and these actually contract and relax to propel food through the digestive tract. And then the serosa is the outermost layer, covers the digestive tract within the peritoneal cavity. The peritoneum helps to allow intestines in the stomach to move freely, yet it also anchors them in place so that they don't get twisted and kinked on each other so that the digestive system can continue to work. Here it shows them all kind of laid out, but they're really actually much closer together. So we have some visceral layers here called the mesenteries. They suspend the digestive organs within the abdominal cavity while anchoring them loosely to the abdominal wall. The mesenteries contain blood vessels, nerves, lymphatic tissues, lymph nodes that all supply the digestive tract. We have the greater omentum is a portion of the mesentery extending from the greater curvature of the stomach and hanging down over the small intestines like an apron. Plays a little bit of a role in the immune system and the infectious process in the abdominal cavity. Then the mesocolon fixes the colon to the posterior abdominal wall. Here we can see the lesser omentum, and that extends from the lesser curvature of the stomach to the liver. So we have the lesser and the greater omentum. Let's move on to talk about the different pieces of the digestive tract. The mouth, also called the buccal cavity. We have the tongue, skeletal muscle covered by mucous membrane, really helps to move food around our mouth to be chewed. The lingual frenulum anchors the tongue to the floor of the mouth. And then we have the hard and the soft palate, which we've kind of already talked about in a previous chapter. And then we have the uvula that hangs down from the soft palate. That's really all you need to pay attention to here. Next, we have the salivary glands. So they secrete saliva, which is a clear fluid consisting mostly of water, but also contains mucus, enzymes that kill bacteria, electrolytes, and 2 different digestive enzymes. Saliva is stimulated by pressure or taste, and we can secrete about 1 liter per day. The rest of the glands here drain saliva, so you don't need to know the specifics about each of them, just that their main job is to drain saliva. The enzymes in the saliva that begin the digestion process are amylase, which breaks down starch, and lipase, which begins the digestion of fat. Really, the main goal of saliva here is to moisten food and transform it into a bolus that can be swallowed easily. The teeth are also in the mouth. They help us to chew our food, a process called mastication. This is really that physical digestion that we talked about, or mechanical digestion. We usually have about 32 permanent teeth. They're all designed in their own way to cut, tear, and grind food. Next is the pharynx, which we've already talked about in a previous chapter. Really, it's just a connection between the mouth and the esophagus. But after the food has been broken down, the tongue manipulates the bolus to the back of the oral cavity. The soft palate lifts, closing off the nasopharynx. The larynx rises, forcing the epiglottis over the entrance to the trachea. The food moves through the oropharynx and the laryngopharynx on its way to the esophagus. Remember, the nasopharynx is just for air. The esophagus connects the pharynx to the stomach. It's about 10 inches in length. There's a muscular sphincter called the lower esophageal sphincter at the bottom of the esophagus, and that helps to prevent the backflow of stomach acid into the esophagus. The esophagus lies posterior to the trachea, and then there's glands within the wall of the esophagus that secrete mucus that helps lubricate the food bolus as it passes through. When the bolus enters the esophagus, it triggers wave-like muscular contractions known as peristalsis that help to propel the food toward the stomach. Next, the food bolus is going to reach the stomach. Okay, this is a muscular sac whose primary function is to store food. Also helps to prepare food for digestion, most of which occurs in the intestines. It contracts and churns to break down food into small particles and to help it mix with gastric juices. This results in this semi-fluid mixture called chyme. Chyme leaves the stomach and enters the duodenum by passing through the pyloric sphincter. The normal curvature of the stomach creates two anatomical landmarks: the lesser curvature along the upper portion of the stomach and the greater curvature along the lower surface. And the stomach is divided into four regions: the cardia, the fundus, the body, and the pylorus. The stomach contains the same layers as the rest of the digestive tract. However, the muscularis layer also has an extra layer of oblique muscle, and that's what helps it to really contract and churn to break the food down into that chyme. When the stomach is empty, the mucosa and submucosa wrinkle into folds called rugae, kind of like the bladder we talked about last chapter. As the stomach fills with food, the rugae flatten and the stomach expands. Empty stomach has a volume of about 50 mils, and a full stomach can contain 1 to 1.5 liters. So it has a pretty great capacity to stretch and become enlarged. Inside the layers of the stomach, the gastric mucosa contains depressions called gastric pits. Several different glands called gastric glands open into the bottom of each gastric pit. These glands produce the various components of gastric juice. We can produce up to 3 liters per day of gastric juice. Mucus cells secrete mucus, which protects the stomach lining and keeps the stomach from digesting itself, so a protective mechanism. Parietal cells secrete hydrochloric acid and intrinsic factor. Intrinsic factor is necessary for absorption of vitamin B12, and a hydrochloric acid helps to kill microbes in the swallowed food and makes our stomach more acidic of an environment. Chief cells secrete digestive enzymes, and enteroendocrine cells secrete the hormone ghrelin, which secretes or stimulates the hypothalamus to increase appetite. It also secretes gastrin, which influences digestive function. So many things happening there in the layers of the stomach. Swallowing signals the stomach to relax so food can enter. Once the food enters, the following steps occur. Contractions originate in the fundus and move toward the pyloric region. They start out gently and progressively become stronger. They're wave-like peristaltic contractions. They help to break down food and help it mix with the gastric juices to form the chyme. The chyme moves towards the pyloric sphincter, propelled by these contractions. Under the pressure of the contractions, about 30 mils of chyme can squirt into the duodenum past the pyloric sphincter. The stomach continues to churn and mix until all of the chyme is released into the duodenum. About 4 to 6 hours, depending on how liquidy the meal is, how solid it is, and how fatty it is. Fatty meals take longer time to turn into chyme and to be moved out of the stomach. Next, we're going to talk about the liver. So although it is an accessory organ, it still plays a hugely important role in digestion and a big role throughout our body, actually. The liver performs over 250 tasks. Things like storing and releasing glucose, processing vitamins and minerals, filtering toxins, recycling old blood cells. It's really the body's largest glands. It has 4 lobes. The right and left are visible from the anterior view. They're separated by the falciform ligament, which serves to anchor the liver to the abdominal wall. The other 2 lobes, the caudate lobe near the inferior vena cava and the quadrate lobe next to the gallbladder, are visible from the posterior view. The portal vein carries oxygen-poor but nutrient-rich blood from the digestive organs and spleen to the liver. So, oxygen-poor, nutrient-rich, because it's getting all of its nutrients from the digestive system, but it's already returning from the capillary bed, so it's oxygen-poor. The hepatic artery delivers oxygenated blood from the aorta to the liver. So the liver is dealing with both oxygenated and deoxygenated blood. There's the liver broken down a little bit more. The liver has tiny 6-sided cylinders called hepatic lobules. They fill the interior of the liver. They're the functional units of the liver. Okay. A central vein passes through the core of each lobule. Sheets of hepatic cells called hepatocytes fan out from the center of the lobule. And in between these sheets are passageways filled with the blood, and they're called sinusoids. Okay, so you can see those there on that picture. See how it's kind of fanning out from the center. Then there's tiny canals called canalicules, and they carry bile secreted by the hepatocytes. So lots of things happening in the liver. And we're going to talk more about how all these things kind of happen or what they all kind of do here. So this is even a kind of more zoomed in look at the liver. So what happens is nutrient-rich blood from the stomach and intestines enters the lobule through the small branches of the portal vein. Oxygen-rich blood enters the lobule through small branches of the hepatic artery. The blood filters through the sinusoids, allowing the cells to remove nutrients as well as hormones, toxins, and drugs. At the same time, the liver secretes substances such as clotting factor, albumin, angiotensinogen, and glucose into the blood for distribution throughout the body. We have phagocytic cells called Kupffer cells. They remove bacteria, worn out red blood cells, and debris from the bloodstream. The central vein carries the processed blood out of the liver, and the canalicules carry the bile secreted by the hepatic cells and ultimately drain into the right and left hepatic ducts. So lots and lots of things happening here. This is where we're cleaning the blood, and the liver is also secreting things into the blood to be distributed to the rest of the body. The gallbladder is attached to the underside of the liver, stores and concentrates bile. It's about 3 to 4 inches long and holds 30 to 50 mils of bile. The liver actually secretes the bile, and so then the gallbladder can store it. The liver can secrete up to 1 liter of bile per day. Bile really contains 2 major things: bilirubin, which results from the breakdown of hemoglobin. They also contain bile salts, which is the main function in digestion because it aids in digestion and absorption of fats in the small intestine. So the bile does have a job. The bile leaves the liver by the right and left hepatic ducts. These 2 ducts converge to form the common hepatic duct, which goes on to become the common bile duct. Bile from the liver first fills the common bile duct before backing up into the gallbladder through the cystic duct. The bile duct merges with the duct of the pancreas to form the hepatopancreatic ampule. The ampule enters the duodenum at a raised area called the major duodenal papilla. And there's a sphincter there. The sphincter of Oddi controls the flow of bile and pancreatic juices into the duodenum because, again, they play a role in digestion. People can live without their gallbladder. Sometimes they have issues with secreting the right amount of bile. but we still can secrete bile and break down fats even without our gallbladder. Next is the pancreas. The pancreas, we kind of already talked about, has both endocrine and exocrine functions, okay? Most of the pancreas consists of exocrine tissues. Each day it secretes about 1.5 liters of pancreatic juices. Essentially, they are digestive enzymes in an alkaline fluid, and they secrete those into the small intestine, right into the duodenum. We have the acinar cells that secrete the digestive enzymes in an inactive form. Once activated in the duodenum, the enzymes help break down lipids, proteins, and carbohydrates. So huge process in digestion. The duct cells secrete sodium bicarbonate, which helps buffer the highly acidic chyme entering the duodenum from the stomach. So we don't want that to be really acidic in our intestines. The ducts converge on the main pancreatic duct, which joins the bile duct at the hepatic pancreatic ampulla to be secreted into the duodenum. Next, we have the small intestine. It's called the small intestine because it has a smaller diameter than the large intestine, but it is actually much longer than the large intestine. But that's why it's called the small intestine, because it's smaller in diameter. Most chemical digestion and nutrient absorption occurs here in the small intestine. The duodenum is the first 10 inches. It begins at the pyloric valve and ends as the intestine turns downward. It receives chyme from the stomach, as well as pancreatic juice and bile. This is where stomach acid is neutralized and pancreatic enzymes begin chemical digestion. Most digestive processes occur here in the duodenum. That's that green part on the picture there. The next part is the jejunum. It's about the next 8 feet. Many large, closely spaced folds and millions of microscopic projections give the jejunum an enormous surface area, making it an ideal location for nutrient absorption. Okay. The ileum is the last 12 feet. The wall of the ileum is thinner and has less blood. Clusters of lymphatic nodules called Peyer's patches are scattered throughout the ileum. Here is showing you 2 different processes that occur in the small intestine, segmentation and peristalsis. Segmentation involves ring-like constrictions that occur at intervals along the small intestine. When the first contracted areas relax, new contractions occur in other locations, and this back and forth movement breaks up food particles and kneads the contents of the intestines, mixing it with digestive juices, helping it to break down. Peristalsis begins once most of the nutrients have been absorbed and undigested residue remains. We get these wave-like ripples moving the digested material along the gastrointestinal tract. And this is the muscular layers of the digestive tract working to propel that food bolus along. The intestinal lining contains multiple projections that increase its surface area and enhance the absorption of nutrients. First, we have the circular folds that slow the progress of chyme and increase its contact with the mucosa. We want that to happen so that it's sitting there so we can absorb what we need of the chyme. On top of the circular folds are projections called villi. Okay, so here you can see these little villi projections. They really increase the surface area of the small intestines. They help to increase absorption. They're covered by absorptive epithelial cells and mucus-secreting goblet cells. An arteriole, a venule, and a lymph vessel fill the core of each villus. The epithelial cells covering the villi have a brush border of ultrafine microvilli. These, again, increase the absorptive area and produce digestive enzymes. So this is where actual absorption of nutrients is occurring, right here on these little villi. So now that we've talked about kind of the anatomy pieces, let's talk about a little bit more of the physiology and what really is kind of happening during chemical digestion and absorption. So it uses digestive enzymes. We talked about some of them so far. We're going to talk about more. Helps to transform food molecules into particles that can be absorbed. Most of this occurs in the small intestine, and the processes vary between nutrients. So we're going to talk about carbs, proteins, and fats all separately because each process is slightly different. Carbohydrate digestion begins in the mouth with amylase. Remember that carbohydrates consist of sugars called saccharides. So we have polysaccharides, disaccharides, and monosaccharides. Most food contains polysaccharides, which must be broken down into smaller particles. So the salivary glands secrete the enzyme amylase, which helps to break down polysaccharides into disaccharides. Then the low pH of the stomach acid inactivates the salivary amylase. And in the small intestine, when chyme mixes with the pancreatic amylase, starch digestion resumes. So no real carbohydrate digestion happening in the stomach. And the final steps occur at the level of the villi. The membranes of the epithelial cells covering the villi contain the enzymes sucrase, lactase, and maltase. As chyme slides against the brush border, the enzymes bind with the disaccharides. And the final step in digestion process produces glucose, which is immediately absorbed. So we really have to use multiple enzymes here to break down those polysaccharides to glucose, so glucose can ultimately be absorbed into our bloodstream. Next is protein digestion. Begins in the stomach with pepsin. The body cannot absorb amino acids. It must break the peptide bonds, which it does with enzymes called protease. They're absent from saliva. Protease work in the stomach and the small intestine. The enzyme pepsin in the stomach breaks down the peptide bonds between certain amino acids. Then when the chyme enters the duodenum, pepsin is inactivated, but then we get pancreatic enzymes, trypsin, chymotrypsin, and they assume the task of breaking apart the peptide bond. So first it's pepsin, trypsin, chymotrypsin, are pancreatic enzymes that kind of break them down further. Then once it gets into the small intestine, it again comes in contact with the villi, and we have peptidase break down the remaining amino acids into individual amino acids, and then we can be absorbed into the bloodstream. So we have to get the proteins broken down into individual amino acids so that we can absorb them and then reutilize them. Last but not least is fat digestion. When a fat globule enters the duodenum, two substances in bile, lectin and bile salts, break up the fat into small droplets called emulsification. Once emulsified, pancreatic lipase begins to digest the fats. Fats are broken down into a mixture of glycerol, short-chain fatty acids, long-chain fatty acids, and monoglycerides. The glycerol and the short-chain fatty acids are absorbed into the bloodstream of the villi. Long-chain fatty acids and monoglycerides, which are too large to be absorbed by the capillaries, are absorbed into the fatty walls of the villi. So again, just another step we break down. Most of this is happening by the pancreatic enzyme lipase and, of course, bile salts, which we had mentioned earlier. The last part is the large intestine. So really, once food has been processed and nutrients absorbed, the remaining residue enters the large intestine, which absorbs large amounts of water before passing the resulting material, which will become feces or stool, out of the body. The large intestine begins in a blind pouch called the cecum. Attached to the lower end of the cecum is the appendix. The appendix contains masses of lymphoid tissue and serves as a source for immune cells, but we can certainly live without our appendix. At the point where the ileum meets the large intestine is the ileocecal valve, which helps ensure that material moves in a forward direction only. The ascending colon extends up to the liver. Colon makes a sharp turn at the right hepatic flexure. The transverse colon passes below the liver, stomach, and spleen. The colon turns sharply downward at the left splenic flexure. The descending colon extends downward along the left side of the abdominal cavity, becoming the sigmoid colon, which forms an S shape down to the rectum. The rectum is the last 7 to 8 inches of the intestine. The anal canal is about the last inch of the rectum, opens to the exterior through the anus. We have two sphincters here, an internal sphincter and an external sphincter. Again, the internal sphincter is involuntary. External sphincter is voluntary for those of us that have control of our bowels. Let's watch the one animation that is associated with this chapter.
The digestive system performs the vital task of transforming food into chemicals that cells can absorb and use for energy. The digestive system breaks down food into its simplest components. It then absorbs these components so they can be distributed throughout the body. The process of digestion begins in the mouth, where food is broken into pieces. Saliva, secreted by the salivary glands, moistens the food and transforms it into a mass called a bolus, that can be swallowed easily. Enzymes in the saliva begin the breakdown of starch and fat. As the bolus is swallowed, the soft palate lifts to close off the nasopharynx, as the epiglottis covers the entrance to the trachea. Food moves through the oropharynx and laryngopharynx and into esophagus. The bolus travels down the esophagus and enters the stomach, a muscular sac whose primary function is to store food. The stomach also prepares food for digestion. The stomach contracts and churns to break food into small particles and to mix it with gastric juice, creating a semi-fluid mixture called chyme. Chyme leaves the stomach through the pyloric sphincter and enters the duodenum of the small intestine. Bile from the gallbladder and digestive enzymes from the pancreas enter the duodenum to aid in digestion. Muscular contractions called peristalsis propel the chyme through the other parts of the small intestine, specifically the jejunum and the ileum. Most nutrients are absorbed in the small intestine. The residue remaining after most nutrients have been absorbed moves into the large intestine. The large intestine begins in a blind pouch called the cecum. The parts of the large intestine are the ascending colon, transverse colon, descending colon, sigmoid colon, and rectum. The undigested residue moves through the large intestine, where large amounts of water are absorbed. The resulting waste, called feces, passes out of the body through the anus.
So that was a good review of what we just talked about, and that ends our lecture for today.